but some enzyme can do mono di as well. Basically, in this, I know suvathrin and IH1, it do trimethylation for sure, but dimethylation could be there. If you remove this one, it will lose the K9 trimethylation. For example, I have studied a lot about K36. I have studied little bit about this, but my work only on K36 trimethylation. So I know that in K36 trimethylation, to extrapolate your question, set D2. This is the enzyme which only do K36 trimethylation. So if you knock out set D2, then in the cells you have only K36 dimethylation. So this enzyme, NSD1, NSD2, it can do both K36 mono and K36 di. And these are all enzyme important in cancer and other feature. This one, the other thing, some can do only di to tri. Okay, so that's why this is the division of function in the cells, and that's why a lot of enzymes are there. So this setmar, this enzyme is important in DNA damage response pathway because it does di to tri. So honestly, there is a lot of literature available. So, but in yeast, if you do not have a set2, you will not have a K36 monomethylation, you will not have a K36 dimethylation, you do not have a K36 trimethylation. Okay, so these are the examples. Okay, so you understood the concept. I just wanted to put you concept because when you read, you at least you know that how to approach the problem. Okay. So this is this. Now the second thing, so you can design based on that reader activity also. So mostly the numerous chromatin associated factors, they have either chromodomain or they have a Tudor domain or MBT domain, PhD finger domain, bromo domain and 1433. So these domains should be there in the proteins. So why these are important? Because they bind certain marks. For example, this enzyme has this chromodomain, so it will bind K36. This enzyme, this has a Tudor domain. From the color coding, you can see they will bind this one. So the point I am trying to make that usually these are called the reader protein. And as far as my general concept is there, if you target reader protein, it is easy. Why? Because reader protein which perform the function, reader protein actually attracts other factors. So think about in a transcription. In transcription, when you need to start the RNA production, so there is an initiation state and there is an elongation state. So once you've done the modification, this modification are recognized by different factors, and that factors actually provide the context. Okay, so what next thing has to be performed in the pathway is actually determined by this protein. So if you know the context, you can come up with the treatment. And that's why one example I'll give you is bromodomain inhibitor, which is mostly in the cancer field. So bromodomain protein bind acetyl mark, and then once this bind to acetyl marks, it recruits other factors. Now this domain will also found along with the reader protein. So reader protein has a methyl transferase activity, but sometimes they have also this domain. So it will methylate as well as bind it. So these are the different examples. So reader proteins are the main player. Okay, and we'll see some examples that is there. So the second important thing, and the only thing I am teaching which is very well established in the field of epigenetics, I am not teaching those which is there, but same kind of concept has been applied. Now before going to this complicated slide, I will do again that practical thing. I told you that heterochromatin provide the repressive mark, right? So during cell division, next is you have to remove the heterochromatin, right? So how you can remove it? Or in facultative heterochromatin, some stage the gene will be silenced, it will get the environmental signal, it start expressing. So how will you transition from repressive environment to expressive environment? Means transcription to no transcription, or no transcription to transcription. How will you going to do that? So this is going to solve the problem. These are the from three you will find. So let's focus on any example. You can say K9. Let's focus on the K9. So K9 trimethylation is there, transcription is off. So if I want to make the locus active, what will I do? We can erase the K9 methylation. Perfect. So how will you erase the K9 methylation? Erasers are there. Now why erasers will come and quickly remove the K9 methylation? It will provide some signal. Okay. So what kind of, how K9 trimethylation can removed? By using, say, demethylase. So the important concept is why a demethylase has to act. What is the specificity? I told you this answer yesterday. So you understood the problem, right? The problem is a locus where K9 trimethylation was there, but we need to make it acetylated or try to make it remove it. Then we need the chromatin modifying enzyme. You said demethylase. So most of the time this chromatin modifier acts through the complexes. So that complexes where the complex could associate with the other proteins. So it is not a one player game. It is a multiplayer game. And how to probe the complexes? Different dyes you will use, then dye will against what?
Bind particular eraser and see how it travels inside.
So eraser will be a protein, right? So why that dye will not bind another protein? So you said that by immunofluorescence. Okay. So you need to tag two different protein with two different immunofluorescent dye. Then how will you visualize it?
sorry, in the heterochromatin region, it will acting in heterochromatin region.
Yes, you can do that. You can follow the marks. And what other way it can do? What other way it can do? You can do co-IP experiment. If you want to show biochemistry, you can do co-IP experiment. You can pull down the complex by co-immunoprecipitation experiment. So you tag the demethylase with some flag or V5. So those who are not understanding what I'm trying to tell, that any protein in vivo, if you want to study, you can put a tag. Tag means some domain or something which you can specifically recognize. Flag is the one people use. V5 tag are there, HA tags are there. So these tag you can put on there and then you can recognize it. Let me take this. Maybe this is okay, it's gone. So you tag against flag and then you do the flag purification and do the immunoprecipitation. Okay, so you can do that. So that's where you can prove. Other way is your this one, like a serine 10 phosphorylation. So if you do the cheap experiment with serine 10 phosphorylation, and if you pull down or do cheap experiment with K9 methylation experiment in a cell, you know the locus, you know all cheap experiment, right? I'm telling you that I have to prove that when K9 trimethylation is involved in heterochromatin, but serine 10 phosphorylation is involved in euchromatin. I'm giving this statement to you. This you have to remember, right? If you do not remember this, then you cannot pass the exam. But you are sitting in the interview. I'm asking this question. How will you show it? You have an antibody against K9 trimethylation. You have an antibody against S10 phosphorylation. How will you show you?
Where it is locating. If it is locating to the center of nucleus, we can say it is chromatin region.
So you can do that, but in some cases the signal could be in both are very close, right? How will you distinguish? Immunofluorescence, if you are doing see the residue number, K9 is also there, S10 is also there. So what you are expecting? Just tell me if you do the immunofluorescence experiment.
S10 is we are saying S10 modification is seen in your chromatic region. We can tag the immunofluorescence with a red color. If you
say serine 10 is red and K9 is green. Now what will your expectation? What will you see?
Observe it under any microscopic study like the confocal microscope, and we can see where red color appear and where green color appear, and we can say the K9 modification is seen in heterochromatin. So red region, green region depletes heterochromatin, and red depletes euchromatin.
So you are saying that you will see red region and you can see the green region. But when I did experiment, I find both. So your name?
Because
Vikas. Again quiz. How many of you agree with Vikas? And other people are not agreeing. Why? See, you have to interact. This is the learning information. This is there. But my point is, how will you design the experiment? Designing experiment theoretically knowing is the important thing, right? He is saying that when K9 red is there, sorry, K9 was green. Green is there is no red. Red is there is no green. And few people are agree. I told him that both could be there. और some other people will say that sir कुछ भी नहीं दिखेगा वहां पे बेकार लोकस है.
so both कैसे दिखते हैं? Either the chromatin will be in the
I'm asking. I don't know. Both दिखेगा कि नहीं दिखेगा? How many of you agree that both can happen? Both can happen? Raise hands. How many of you think both can happen? Okay. So why both will happen?
Both heterochromatin and euchromatin regions can lie intertwisted.
Fair enough. Agree. So आप कह रहे हो कि ट्रांजिशन स्टेट में हो सकता है K9 कुछ पॉपुलेशन में K9 भी हो सकता है, कुछ पॉपुलेशन में S10 भी हो सकता है. क्या करेंगे आप? देखिए उनका तो आर्गुमेंट वैलिड है ना. Okay, so ideally he is also right, that is also right. You have to do the experiment basically. But yes.
Some enzymes which can specifically that like if acetylation is going on, we can do the acetylation for them. So likewise, we can that only the targeted sequence, like if methylation is our target, so we can take the methylation and the acetylation will all be with that.
Okay, so it's, you are basically adding another complexity. She is basically saying that, sir, you probe this, but along with this, you probe other thing also, so that you will have some confidence. I like that approach also, but he is also right, and other explanations are also right. Then you have to intelligently choose the regions. If you are probing heterochromatin, you have to know that heterochromatin will be there at all the stages. Then if you will only get the green signal. If you know that euchromatin will be there, then only you will get the serine 10. But there could be region where both can coexist. You don't know the dynamics, because I said that chromatin is not a packaging material. It is a very dynamic stage and it acts as a signaling molecule. So both of the situation ideally possible. But actually if you do the experiment, if the cells are in mitotic phase, you will do the serine 10 phosphorylation. The nucleosomes isolated from mitotically active cells, you will see very high serine 10 phosphorylation. No K9 trimethylation. This is known in the literature. But if you are taking interface cells, then it could be both this and this, because it is a population of the cells. So clear ho gaya na? In this way, you can say that serine 10 phosphorylation actually inhibit K9 methylation, but serine 10 phosphorylation actually activate K14 acetylation, because K14 acetylation is involved with the active transcription. So ideally, one modification can cross-talk with the other modification. And that is makes the chromatin very complex and complicated because it acts in a crosstalk way. And why crosstalk is necessary? Crosstalk is necessary to provide the functionality, because I told you that epigenetics is the stable, inheritable changes without DNA sequence. So one thing might not, these are the very strong signal like K9, serine 10, K27, K36, they are abundant. So initial days people have quickly probe it and figure it out that one corresponds to the other. But actually in the cell it acts in a context manner. So similarly, they can be different type of situations. So designing inhibitors against K9 trimethylation or K27 trimethylation or K36 trimethylation will be very, very difficult because they are acting in different processes. Okay, so these are the same thing. This is other examples are also are not going in detail because this you should know. Now let's focus about histone acetylation, because this is important so that we can know what you require. So if you want to design a drug specific to the acetylation, you have to focus on the enzymes which do the histone acetyl transferase. You need the acetyl CoA and also enzyme which remove these modifications. So you have to either focus on HAT or HDAX, and then to bind this modification you have a reader proteins. And in the reader proteins you have a different classes of modification, and the most of them which is known in the literature is for BRD4 and BRD2 inhibitors. Those inhibitors had been gone to till the clinical trials, and I'll come to the name of the inhibitor in the later on. So if you want to come up with the new inhibitor, first what the stage you have to do, you have to figure it out that which methyl transferase or which epigenetic marker is involved in the disease. Now your next stage you figure it out which enzymes, and then you have to figure it out to bind the modification is there is something else or not. So you can target from different way. Okay, so your target could be any of these things. You can target HAT, you can target HDAX, you can target this one. This is the simplest experiment. You can do the enzymology, you can do the biochemistry, and then you can come with the chromatin inhibitors. Now you are wondering that why this is so many classes, right? दे एक कुछ नहीं है, क्यों है? एक तो ऐसे ही आंसर कर सकते हो कि दे आर डिफरेंट प्रोटीन्स, दे विल हैव सर्टेन डोमेन्स विल बी देयर नॉट देयर. अदर थिंग इज context specific. So different bromodomain proteins, they do not act in isolation. It is not that there is acetylation in the cell and bromodomain is binding. That bromodomain content protein has to come with the other complexes. So what different bromodomain proteins are coming with the different complexes, and that's why they are giving you different function. So some will be involved in the one process, other will be in other process. So this is the simple example. If you treat the cells with sodium butyrate, it will inhibit the HDAC function. And these are the other things. So basically, I am going in very depth now. So the depth is you have a lysine side chain, and that is the amine group where acetyl group is added. Those who like the biochemistry, since I have to keep this for all of you. So I am putting all things. I put biochemistry also, I put cell biology also, I put immunofluorescence also, so that I can at least tell whoever, somebody will be interested in this. मुझे सर वो कॉम्प्लेक्स सेल बायोलॉजी नहीं करनी है, मुझे बायोकेमिस्ट्री करना है. Then you have to go to the residue level, which marks and which enzymes. So you learn now how to make nucleosomes. You learn now how to get the enzyme, HDAX and this. You can co-incubate. You can do the biochemistry. Do you need to do the fancy seq experiment now? No. If you come up with a novel chromatin modifier as your thesis, or you can do just biochemistry. If you show any of this on new things, who cares? That you don't need to learn epigenetics. Simply, I promise that I will teach cell biology and biochemistry at the end of the day, so that from your brain that epigenetics हवा को निकलना चाहिए कि ये तो कोई ऐसा चीज पढ़ा रहे हैं ना, ताकि मार्केटिंग काफी हो रही है कि epigenetics is such a new area, such a brand. You know, we all love brand, right? So come out from the brand, because never know who. No, you could come up with a protein which is not known in as a histone modifier, histone modification. It is a non-histone target. So many times now people are wondering that why hell in the mammalian cell these different methyltransferases are there. They could have a non-histone target also. So don't come with a pre-mindset that histones could only be the target. The substrate could be non-histone also. There also your approach could be this. And now people are coming with the different non-histone products. P53 is acetylated. People have come. People have also shown many examples. Clear? This is just show you to make your life more complicated, because for HDAC and HATs, if you read the literature, it is everywhere. Okay, people have shown that HAT can work on non-histone target like P53, RB, greater than 60 proteins. HAT could be done by P300, PCAF, HDAC. It could be involved in RNA. It could be involved in metabolic. But I hope that I told you everything. The reason I'm saying this, because I told you yesterday that many times the cofactors of the enzymes are metabolites. So I am not wonder that some metabolic enzymes can affect the activity of this. So now if you think the problem conceptual, if you go to memorize this, your life is going to be difficult. But I'm telling you not to memorize. I'm telling you the problem. The problem, I started this class telling that to access the DNA is our aim. Either मुझे DNA को बाकी सेल के मशीनरी को एक्सपोज करना है या उससे बचाना है, यही काम है. तो जैसे मैं कह रहा हूं कि if acetylation is important for replication, so can you come and explain to me in a very naive language why it is important for replication? Why acetylation is important for replication? मैंने कुछ पढ़ा नहीं है, मुझे बस बता दो कि I can explain this just by knowing how to access the DNA. How will you explain it? Replication and transcription. इस पे पॉज करते हैं थोड़ी देर, 5 minutes to think through.
It will recruit some proteins which will be helpful in replication or transcription. Right?
Very good. It can recruit some proteins through the reader domain because acetylation is important. मैंने बोला वो तो आपने ऐसे ही पढ़ के बोल दिया, मुझे कुछ और चाहिए. और क्या ऐड कर सकते हैं इनके आंसर में?
release the complexity of the so that application is easy.
Complexity of chromatin in what context? ये थोड़ा सा क्लियर नहीं हो रहा है.
Like the DNA moves out because histone is a positively charged, it can induce some negative charge and make loosening of DNA. So DNA will be active.
इधर से तो कुछ आंसर ही नहीं आ रहा है और पीछे से कुछ नहीं आ रहा है.
Don't जो पॉजिटिव चार्ज होता है
हम्म.
उसको कम कर डीएनए का जो नेगेटिव चार्ज और अस्टाइलेशन जो है वो पॉजिटिव चार्ज जो होता है वो कम करता है. तो नेगेटिव और पॉजिटिव का जो इंटरेक्शन है वो कम होता है, वो लूजनिंग होता है.
ओके, वो हेल्प करता है रेप्लिकेशन में, यही आप कहना चाहते हो? पीछे ये लोग सही बोल रहे हैं दोनों? कितने लोग सोच रहे हैं कि ये दोनों सही बोल रहे हैं? नहीं, एक बार हाथ उठा के रखिए. सॉरी, आई एम नॉट पनिशिंग एनीवन. जी, जो हाथ नहीं उठाए हैं बहुत कम लोग हैं. ठीक है, सभी ने हाथ उठाया है. किसी ने हाथ नहीं उठाया है तो बोलो. मैं बोलने वाला हूं, इनका आंसर गलत है. कितने लोग बोलते हैं इनका आंसर गलत है? हाथ उठाइए. इन्फ्लुएंस नहीं होना है कि सभी हाथ उठाए तो आंसर सही होगा. मैं बोल दूंगा गलत आंसर है. कुछ लोग हैं जो गलत आंसर समझते हैं, इसको गलत है. मैं उठाऊं हाथ? मैं तो उठा ही सकता हूं हाथ. मैंने 3 नंबर का क्वेश्चन दिया, बोला कि मुझे बताओ, तो आपने आंसर किया. पहला आंसर किया कि कुछ क्रोमैटिन कॉम्प्लेक्सेस कैन बी गेट रिक्रूटेड जो रेप्लिकेशन करते हैं. फिर कुछ ने आप दोनों का आंसर लिखा. मैं सेटिस्फाई नहीं, मैं नहीं से 5 में 5 देना ही नहीं है मुझे, इसको 3 ही दूंगा. कुछ और भी फैक्टर है जो मुझे चाहिए. हाँ,
सर जब भी ये लाइसिन में एसिटाइलेशन कर रहा है तो मे बी कि डीएनए रेप्लिकेशन प्रमोट हो सकता है, पर डिफरेंट अमाइनो एसिड में एसिटाइलेशन करने की वजह से ये प्रमोट नहीं भी तो कर सकते.
ठीक है, थॉट प्रोसेस ठीक है, हो सकता है सर. आइडियली थ्योरेटिकल. नाइस अप्रोच. ऐसा हो सकता है और ऐसा इनफैक्ट होता है मेनी टाइम्स. 2 चीजें हैं एपिजेनेटिक्स एंड क्रोमैटिन में. वन, यू गो फॉर ए जनरल description, another go for context specific, what she was talking. So this is again my daughter time pickup. So बिल्कुल हो सकता है, but I agree with all of you that from the problem of in replication you need to duplicate the DNA, and replication machinery only can make a copy of DNA if it need to access. So if you put the acetylation, then that will loosen the, and then there will a protein which will basically dismantle. But what she is saying, she could be right. Many times it happens. Okay, but the general, I agree with this. At least for one part, everyone got agree. Okay, so now I think I'm happy that this is the good class. I said that initially response was very low, but now in the last we have like a half an hour class left, 20 minutes left maybe. Everyone at least agree on one thing, which is a very good thing. I love it. Okay, so acetylation we lose, so you can just explain this. Same transcription को भी explain कर सकते हैं. Our purpose is to access the DNA. So acetylation where you can reduce the charge and then you can access the DNA. And once the thing has been done, you put back HDAX. Okay, enough transcription has been done. This locus has to be shut down, so it will remove the acetylation. So that's why this one is a very well-known study. But if you come for STK36 methylation, वहां इनके तरह situation हो जाएगा, because that modification you cannot explain by access. So the